Alright, so today we're going to be focusing on the electron transport chain and oxidative phosphorylation. Okay, so these are the processes that we will use to somehow convert NADH and FADH2 to essentially ATP. Okay, so what we're going to do is we will take, remember this is TCA cycle, okay, and remember that in TCA cycle you will generate a bunch of electrons, okay, all these electrons, okay. So all of these electrons will be used to do something, okay, and today we're going to be talking about respiration, okay. So this is the process that you use when you breathe in O2 and you put the electrons, okay, so the electrons onto molecular oxygen, okay. So you are going to reduce molecular oxygen into water, okay, and in the process what you're doing is essentially this, okay, converting ADP plus PI into ATP. Okay, so this is the one of the most important processes that will occur in your mitochondria. Okay, remember back when I think it was like what, middle school, high school, you learned about mitochondria, okay, and you learn about how mitochondria is the powerhouse of your cell. Okay, so mitochondria is used to generate energy. What is energy? You know, ATP, etc. Okay, so electron transport chain. Okay, this is what we'll be talking about. Okay, this part. So we finished talking about PDH. We finished talking about TCA. Now we're going to be talking about the electron transport chain, or sometimes people just call it ETC. Okay, 不是那个高速公路要用的东西哦。Okay, 是 ETC. Okay, electron transport chain. All right. Here are some of the central questions that we will ask. Okay, one is how do cells utilize NADH and FADH2 to make ATP? How does this process work? Okay. Number two, how many ATP can we make per NADH or per FADH2? Right. And number three, what are the enzymes and metabolites involved? Okay, so you know, biochemistry. We're here to talk a little bit about what are the enzymes, what are the metabolites involved in this entire process. Okay. Now, before we move on, just to remind you guys that mitochondria is really the powerhouse of your energy generation. Okay, and you should have an image in your head. Okay, let's say you have glucose in your bloodstream. Okay, glucose in your blood, and how does that glucose get converted to CO2? And where do the conversions take process, okay, occurs? Now, let's say, you know, here you have your cell membrane, okay? Let's say this is outside. Here is cytosol, okay? And then cytosol, and then inside is mitochondria, okay? For example, glucose gets transported into the cytosol, okay? So then in the cytosol, it's going to do glycolysis, okay? So glycolysis occurs within the cytoplasm, okay, the cytosol. And then once you get, you know, pyruvate, okay? Pyruvate can then be transported into mitochondria and up in the matrix of mitochondria, okay? Now, I know it's an ugly picture, but mitochondria essentially is two lipid bilayers. Okay, you have an outer membrane, and then you have an intermembrane space, and then you have an inner membrane. Okay, so don't get confused with what we say lipid bilayer. Usually when we say lipid bilayer, this is a bilayer, right? Because you know, essentially you have one layer, two layer. Right? That's what we call lipid bilayer. Okay, now every lipid bilayer makes a membrane. Okay? And for mitochondria, or in general for like E. coli, for example, you have two layers or two membranes. Okay? And you know, if you look at look at this mitochondria. This is cytosol. This is the intermembrane space. I just call it IM, intermembrane space. And then in here is the matrix of mitochondria. Okay? All right, so it's actually 3 layers. 1, 2, 3, like 3 spaces. So what you will notice in the matrix of mitochondria, what happens? TCA cycle, PDH, okay, all of those enzymes that are going to generate you a bunch of NADH, they happen in the matrix. On the inner membrane, okay, on the, this part, the inner membrane, you have a bunch of proteins that are going to be stuck on the membrane. These things are called what? Membrane proteins, integral membrane proteins, things like that, okay? And these enzymes are going to do your ETC, generating ATP, things like that, okay? So there are some key enzymes on the inner membrane that we'll talk about. Okay. So this is probably one of the most important slides to summarize the entire ETC and ATP generation. Okay, so let's take a look. Here in the blue, this is the matrix. Okay, so what that means is that you have pyruvate in here. Okay, so you have pyruvate. Okay, and of course you can also have some other stuff. Pyruvate can get converted to acetyl-CoA, and in the process you kick out one NADH and CO2. Remember? Okay, so NADH comes out. Okay, and then you run the TCA cycle. Okay, and the net process, the net reaction for TCA cycle is it will give you 3 NADH and 1 FADH2. Okay, so it generates a bunch of NADH. Right? So the key is that you generate a bunch of NADH. Okay, now NADH, what happens is that NADH will now passes the electrons, okay, the 2 electrons on NADH, onto this protein called complex 1, okay, called complex 1. And in the process, NADH is now returned to NAD. Okay, so this is, you are oxidizing NADH, okay, and you oxidize NADH back to NAD. In the process, you passes the electron onto complex 1. Now complex 1 can then pass that electron to a electron acceptor in the lipid. Okay, I'll tell you what that molecule in just a minute. And this particular molecule can then interact with, this molecule is coenzyme Q. Never mind, it's right here. Okay, so I'll erase this. Okay, so what complex 1 does is that complex 1 will then pass the electrons from NADH onto this thing called coenzyme Q. Okay, called Q. And coenzyme Q then, floats around in the lipid, and then, it touches complex 3. Okay, and then complex 3 can then take the electrons from Q, put it onto this thing called cytochrome C. Okay, it's a protein that has a heme, and then cytochrome C, just like wanders around, and then until it touches complex 4. Okay, complex 4. Complex 4 then takes the electron from cytochrome C and then put them onto molecular oxygen. You generate water. Okay? Now, in all of these processes, what you see is that you will have a net movement of protons from the matrix moving into the intermembrane space. Okay? So you have proton movement, and then here also proton movement. So what you accomplished is that here you have a bunch of protons. Okay? And here in the matrix you have less. Okay, so there's going to be a change in pH. Okay, so separation of chemical species, essentially. Okay, and as you guys know from general chemistry, any time you have a separation of chemicals, okay, in space, what do you have? You have a chemical potential. Okay, so you have a potential energy that will sort of want to take these protons back to the matrix. So you have a potential energy right there, right? So then you have complex 5. This guy is also called the ATP synthase, which we'll talk about in a little bit. And what that guy does is then it will convert, okay, so the protons can then go through this channel, return to the matrix, okay, and in the process, it will sort of turn this protein around, okay. So what it's actually going to do is you will convert the potential energy into like a little mechanical energy, and then use that energy to store that energy from your potential into ADP to ATP. Okay, so you're doing energy conversion here. Right? So we'll see in maybe, if not today, Tuesday, what this thing will look like. Okay, so it's actually a doorknob that rotates. Okay, every time it rotates, it creates a string, and then that string will then be saved into ATP. 有人不太清楚我要讲什么，没关系啊，待会给你看图。Okay, but whatever. The process, okay. So this thing is called ETC. Electron transport chain, right? Because you're just converting electron from NADH to here, to Q, to cytochrome C, and then eventually to water, okay? So this is the ETC itself, right? And what you should notice is that FADH2, okay, that's generated from TCA cycle, okay? It's using complex 2 that will take the electrons from FAD, pass it to coenzyme Q. Okay, now, complex 2 is independent of complex 1. Okay, so if I ask you what is the electron flow in the ETC, please don't tell me you go from complex 1 to 2 to 3 to 4. No, okay, it's 1, 3, 4 or 2, 3, 4. Alright, we clear? Okay, are we good with that? Alright, so complex 2 is independent of complex 1. Alright, turns out, complex 2, this particular enzyme, is the enzyme in TCA cycle that converts succinate to fumarate. Okay, so complex 2 is the same enzyme in TCA cycle. Okay, so all the other TCA cycle enzymes are soluble enzyme. Okay, they're not membrane bound, they're soluble, with the exception of your SDH, your succinate dehydrogenase. Okay, for converting succinate to fumarate. This guy is a membrane-bound protein. Okay? All right, we're good? All right, now, please remember that you have sort of 3 layers, sort of. You have an aqueous environment in the matrix. You have an organic environment in the membrane, okay, in the inner membrane. And then you have another aqueous environment in the intermembrane space. Okay, so there's a lot of key words or key terms that you guys need to be able to differentiate. Inner membrane, intermembrane space, okay? They're different things, right? Just so you know. Okay, huh? All right. Now, and then there's the question of then how do bacteria generate ATP from NADH? Okay, pretty much the same thing. Okay, so if you look at a bacterial cell, okay, what it is that you also have 2 layers. Okay, you have an inner membrane and an outer membrane. Okay, this is outside the cell, this is inside the cell, okay, cytoplasm. And then for bacteria, they have this thing called the periplasm, and periplasm is equivalent to the intermembrane space of your mitochondria. Right, so pretty much your E. coli, for example, is just like your mitochondria. Okay, so this is probably how we even made mitochondria, a fusion between a bigger cell with a smaller cell. Okay? Right, so this is just bacteria, they use the same mechanism, and the ETC of bacteria is located on this particular, the inner membrane. Okay? It's located like these proteins. All right, since we're talking about electron transferring reactions, we need to sort of also look at their energy change. Okay, and how do we assess energy change of electron passing reactions? Right. So in general, your standard delta G can be written in this way, where standard delta G is equal to minus NF in parentheses. Essentially, this guy is delta E. The standard reduction potential of the acceptor minus the standard reduction potential of the donor. Okay, so essentially this is like, delta E. Okay? And F is nothing more than a constant, Faraday constant, and N is the number of electrons that got passed. Okay? All right? So here's an example. When NADH gives its electrons to O2, forming H2O, forming water, this is essentially the reaction, okay, the net reaction. What is the delta G of this reaction? Okay, well, the standard delta G of this reaction. You go to your standard reduction potential table and you identify the standard reduction potential of these two half reactions. Okay, and you put them down. You have 2 electrons passed, 2 electrons. This guy is a constant. Okay, and you take the E naught of the acceptor, and who's the acceptor in this case? O2. Okay, O2 is the receiving electrons. Okay, so this guy is the O2 minus the standard reduction potential of NADH or NAD, doesn't matter. Okay, it's this guy. So this guy is NAD. Okay, and you convert that, it becomes positive. Okay, and if you do that calculation, what you get is about 220 kilojoule per mole of NADH converted to molecular oxygen. Okay, so NADH donates the electrons from NAD onto molecular oxygen, forming water. This will give you about 200 kilojoules per mole of energy. Okay, that's quite a lot, right? In theory, how many ATP can we make from this? Roughly 7. Okay, roughly 7. Why? Because every ATP is about 30 kilojoules per mole in terms of energy, and if you have about 220 kilojoules per mole, then you can get about 7. That's, of course, the theoretical, if you utilize all of that energy, okay? But obviously we don't, okay? We don't, in reality, we don't do that, okay? You can think about why. Why? What do you think? Why? Because if you use all of that energy, you conserve all of that 220, okay, you will reach equilibrium. Okay, so that's not a favored trait. Okay, remember we talked about that last semester when we were talking about how if you, want to convert glucose into all CO2, right? Remember it can give off like a huge amount of energy, like 1,000 or something. And if you take all of that energy and convert it to ATP, you can get about 50. But in reality, we'll only get 30. Okay, so where is that lost? Okay, remember I said that because if you were to do full energy utilization, okay, then your reaction will not have a minus delta G, right? Then it will be like an equilibrium, so you don't have a driving force for survival. Make sense? All right, if you don't understand that, come to me later. All right. So let's take a closer look at the electron transport chain, right? So we have complex 1, complex 2, complex 3, and complex 4. Complex 5, ATP synthase, we usually leave at the very end because it's not exactly in the ETC because it does not take electrons, so we don't consider that as part of the ETC. All right, so complex 1, it's the name for complex 1 is actually called NADH dehydrogenase because it's doing a dehydrogenation reaction on NADH. So you take out the H2 from NADH and you put them onto essentially coenzyme Q. Oops, coenzyme Q. Okay, so right here, complex 1. And then complex 2 is succinate dehydrogenase, or SDH, which is part of TCA cycle, like I mentioned. And what it does is it will transfer electrons from succinate to CoQ. Okay? And complex 3 is this thing called the cytochrome C CoQ oxidoreductase. Okay? So what that just means is that the two sort of chemical species that this enzyme reacts with are cytochrome C and coenzyme Q. Okay, so cytochrome C, CoQ, oxidoreductase. Okay, so they're doing oxidation reduction reactions on cytochrome C and CoQ. Okay, so you will convert coenzyme Q into cytochrome C, this guy, okay, into cytochrome C. Lastly, you have a cytochrome C oxidase. You're oxidizing cytochrome C by using molecular oxygen, okay, so you turn the electrons on cytochrome C to oxygen, okay? Now, what you should notice from this chart is that inside of each complex, you have these iron thingies looking like in the middle, okay? I'll tell you what they are in a minute. You have FMN or FAD, okay? Right, so these enzymes are quite large, okay? Inside of these enzymes, there are multiple centers for electron passing reactions, okay? So it's not one step going from NADH to coenzyme Q, okay? It's actually multiple steps. You're passing the electrons from, let's say, the surface of the protein and then into the protein and then eventually passes on to coenzyme Q, all right? So what you can see is that within each complex, you will have all of these sort of clusters of electron accepting groups and donating groups, okay? So these are portions of the enzyme that can take electron and donate electron, okay? So as you can see from this particular slide, you have a bunch of irons, okay? You have a bunch of iron. And iron can accept electron and become from Fe3 plus to Fe2 plus, okay? And then when it donates that electron to the downstream partner, it goes from 2 plus back to 3 plus. Okay, so it's that kind of process. Now, what you can see is that in each of the complex, okay, you have a bunch of different iron centers, different iron centers. And each different iron center will have a different standard reduction potential. Okay? And what you should notice, okay, is that each subsequent reduction potential, the energy is usually, or actually I should say, the standard reduction potential is becoming a larger number, right? Which indicates that it likes to be reduced. It's more oxidized. Does that make sense? Okay? So for example, you start with NADH, which is about minus 0.3, okay? And you will get down to, for example, this portion of the iron sulfur cluster in your complex 1 is about minus 0.1. Okay? Of course, what you should notice is that you have some, for example, 340 going to 380, okay, 0.34 to 0.38. You're like, how does that make sense? But again, don't forget, this is standard energy, okay? If you control the concentration, if you control the microenvironment of that electron accepting group, you can change the energy of that particular portion of the iron. Does that make sense? Okay. So what you should notice is that a general trend exists for becoming more positive. Okay, the electron acceptor is more positive. Okay, so eventually you go from NADH, which is minus 0.3, to coenzyme Q, which is 0.04, to cytochrome C, which is 0.23, eventually to oxygen 0.8. Okay, so the electron passing direction is from the more minus to the more positive. Okay? I think that should be pretty straightforward. All right? Now, here I'm going to introduce a couple of key electron transporting players, the key electron mediating species. Okay? We have flavorol proteins. Flavorol proteins are those proteins that bind tightly to FMN and FAD. Okay, so we have seen some of those before, where they have a FAD in their enzyme that can do electron passing reactions. Okay. Now, turns out a lot of times these flavorol proteins, essentially they have this cofactor buried inside of the protein, and you guys should appreciate the fact that proteins are 3D. okay, three-dimensional. And because enzyme is three-dimensional and your FAD is in the middle, okay, you can change slightly what the standard reduction potential of the FAD would be. Okay, you can actually change it. Why? Okay, why? Because you can think of, let's say, you have an FAD here, and then you got some aspartic acids or something, okay, that has a charge, for example. So then that could influence the reduction potential of your FAD. Right? Makes sense. Microenvironment of the enzyme can change the characteristics of a particular species in that enzyme. 有听懂我意思吗 ？Maybe, OK， 就是记不记得像我们之前在讲那个histidine 对吧？我们不是之前在讲 amino acid histidine， 然后说为什么 histidine 很重要，因为它的 pKa 是在6到7左右嘛，对不对？那它为什么很常出现在 enzyme catalysis 的 mechanism 里面 ？Why? OK， 因为它6到7，那它可以随着这个 histidine 附近其他的 amino acid residue 去影响这个 histidine 的 pKa，right？ 所以 enzyme 因为它是3D 的 ，OK， 它是一个3D 结构。那你今天有任何一个 chemical 在中间，那这个 chemical 呢，会因为这个 enzyme 其他的 residue 来影响它的一些 property， 比如说 pKa， 比如说 standard reduction potential。OK， 所以会受到影响。OK， 那会跟它这个东西自己在水里的 property 会有一点不太一样。所以你会受到enzyme 本身的 influence. Make sense? Kinda. I think it should make sense. Okay, now, flavor proteins, okay? These flavin cofactors, FMN and FAD, okay, they're very important, okay? Why? Because they essentially serve as a transformer for 2 electron carrier, NADH, to a 1 electron carrier, which is these iron centers. Okay? For example, you see here, you are trying to get NADH, here is 2 electron carrier, onto iron, which is a 1 electron carrier. Okay? So obviously, you can't just NADH come in, smash 2 electrons to iron only takes 1, and the other one just like magically disappear. No, it can't. It doesn't work. Okay? Electrons cannot be created nor destroyed. Remember that? So you have to have an adapter. Okay, it's like a little transformer that can sort of 2 electrons from NADH onto FMN, for example. This guy can take up to one electron or 2 electron, and every time they pass 1 electron to iron. Okay, so it works like that. So FMN is like a little adapter for converting NADH to iron. Okay? So you have these iron-sulfur cluster proteins. Those are proteins that contain iron-sulfur clusters. Okay? And what they can do is they can transport 1 electron at a time. Lastly, you have coenzyme Q and cytochromes. Okay? And there are different cytochromes. The one that's floating inside the membrane is cytochrome C, but we do have cytochrome B and A that's part of the complexes. All right. Flavor proteins, flavin. Basically, you have this this ring right here, and flavin usually when you eat it, okay, is riboflavin. So this is a vitamin, and essentially what it has is a ribose. Okay, that's attached to these this isoalloxanine. I can't even pronounce that. Okay, so whatever. It's a ribose attached to this. All right, but essentially what it is it's vitamin B2. Okay, so you can go home and tell your mom that, hey, yo, I just learned about what vitamin B2 does. Right? So in our class we talked about B1, now B2. What else? B5 for making coenzyme A, and then B7. Okay. So we talked about all these vitamin Bs. Right, so hopefully, you actually learn some stuff from this class. And riboflavin, vitamin B2. Riboflavin can be activated into FMN, and FMN is the same thing as riboflavin, except you have a phosphate group right here. All right? You can then turn FMN into FAD, okay, by adding this. Okay, so this guy is this guy, and then you add this. Okay, so you have FAD. All right, but whatever. The key the key function or the key portion of the molecule is right here. Okay, it's right here. All right, so flavin, again, it can be reduced by NADH to become FADH2 or FMNH2. And if it's reduced in a reduced form, you will have two hydrogens, one right here and the other one is right here. Okay, two hydrogens. And what you can do is you can donate one electron at a time. Okay, so one electron at a time. For example, you can have a little iron 3+ or something right here. Okay, and come in, take one electron, so you can do a homolytic cleavage. So this guy, this is a okay, you learned this in organic chemistry, homolytic cleavage, where you take one electron and the other electron goes to the other molecule. And you can generate this semi-quinone radical, FADH dot, okay, little dot. So every time you see a little dot right here, that indicates it's a radical. And then obviously this iron can then pass the electrons to some other guy, some other guy. And then you spit out Fe, oops, shouldn't draw that like this. So this essentially becomes Fe2+, okay? Fe2+ passes the electron to this guy, and then you kick out, and then you get Fe3+ again, and then Fe3+ can become, white with a dot. And then you take another electron, and then come out, Fe2+, okay? And then it passes the electron to another guy, and then you become Fe3+. So in cycles. Make sense? Okay. All right. And when you do that, you fully oxidize your FADH2 to become FAD. Okay. So this is how FAD is a transformer or adapter. Okay. But it sort of transforms two electron carrier to one electron carrier. Okay. Iron sulfur clusters. Okay. This is actually a very important and very interesting topic. Okay, so actually it turns out there are quite a bit of enzymes that have iron sulfur clusters. No, not just enzymes associated with the ETC. There's all kinds of enzymes that have these iron sulfur cluster proteins. Iron sulfur clusters. But it's not in the scope of our class to discuss in detail iron sulfur clusters. Okay, so you can actually, there are a lot of people, their entire research focus is on these things. Okay, so that, I'm just very briefly telling you what okay, essentially what they are that you have these irons, okay, iron ions that are going to be chelated by these sulfurs, okay, and sulfurs, where do you get sulfurs? In amino acids, cysteine, okay, so usually these are cysteines, okay, and there are some different arrangements, okay, just for you to know, iron sulfur clusters have different arrangements. For example, you have FeS, Fe2S2, Fe3S4, and the Fe4S4. Okay, so they are just different arrangements for how you can put together a iron sulfur cluster. Okay, so obviously different configuration and different protein microenvironment will change their reduction potential. Okay, so that's the message that I want to get across from me to you, is that different iron sulfur clusters will have different reduction potential. They have different microenvironments. Okay, but nonetheless, for our purpose, is that they accept one electron. Okay, so you can reduce them one electron at a time. Okay, so you go from Fe3 to Fe2. Okay? All right? Coenzyme Q. All right, so this is the guy that floats around in your membrane. Okay? Obviously, by looking at this particular structure, you should probably understand why it happens in the membrane. Okay? Because it's very hydrophobic. Okay? It's very hydrophobic. You have this long-ass chain of hydrophobic tail. Okay? This is coenzyme Q. Let me first give you a little description of coenzyme Q. you have this quinone thing, right? This guy is called quinone or ubiquinone. That's linked to a number of isoprene unit, okay? Isoprene is like that, okay? That's an isoprene, all right? Isoprene is actually very interesting, okay? So isoprene monomer by itself is a gas, okay? And you rarely see it just lying around, okay? Isoprene is a gas, but actually not biologically, industrially, isoprene is a very useful chemical, okay? You can actually make rubbers out of it, okay? For example, polyisoprene, okay? 那它是一个天然的这个 material， 那是树脂啊，也就是那个树你切开来，它里面会留一堆那种东西，有没有？那就是 latex，OK？ 那你知道其实 latex 哦，一个小故事啊，就是说 latex 这种东西呢，它是天然的 ，OK？ 但是呢，有些人会对 latex 过敏 ，OK？ 会对 latex 过敏，这就为什么其实像我们现在实验室手套买都是那个 nitrile gloves， 不是那个 latex glove，OK？ 因为有些人对 latex 过敏，为什么会对 latex 过敏？你知不知道？其实不是对这个 polymer 过敏，是对 latex 里面啊，因为它是植物的 origin 嘛，那植物你把它萃出来的时候，它可能会掺杂一些植物蛋白或者植物的一些分泌物，那有些人会对那个东西过敏，因为其实你要知道，很多人他们会对植物的各式各样的东西过敏，有没有？像花粉啊、树粉啊、一些不同的树的粉啊 ，OK， 所以latex 过敏是因为它里面会有一些东西 ，OK， 所以 as a result， 其实现在很多人在做什么？就像我们 research 哦，就是会有人要做的就是，那我让微生物去合成 isoprene， 我就 isoprene 就好。那 isoprene 是气体，所以你在合成它的过程中，它会被蒸发出来，就是它会出来。然后你出来，你把它 condense 到另外一个地方，那你就有纯的 isoprene。然后你再拿纯的 isoprene 去合成 polyisoprene， 那你就会有一个比较纯的 material。那种东西对过敏体质人来说就比较不是个问题，这样 ，OK， 这是 isoprene， 这是 isoprene。那 polyisoprene 的话，当然就是一整条嘛，一整条这种东西叫 polyisoprene，OK。Now quinone，OK，this thing is called quinone or ubiquinone，OK，ubiquinone，UB 有没有知道那个 UBI 或是 ubiquit 这个东西是这个字源，这个字是什么意思？有没有听过一个英文字叫 ubiquitous？ 就是 everywhere. Okay, so it's everywhere. So this kind of molecule was initially named for its presence in almost everywhere. Okay, so it's called ubiquinone. All right, so ubiquinone, why it's called quinone? Okay, is because you have ketones right here. Okay, so ketone, and you can of course reduce it one at a time. Okay, so you take one electron and you will generate this UB semiquinone. So you can write it as QH with a little dot, QH radical. Okay, so the radical is right here. So one of the O becomes alcohol and the other one is a radical. You take another electron, okay, then you have another, it becomes alcohol. Okay, so this guy is ubiquinol, no, for alcohol. Okay, so this is alcohol. Okay, so oh yeah, by the way, ubiquinone CoQ, usually for us in mammalian cells is Q10 because it has 10 isoprenes. Whatever lah. Okay, so 10 isoprenes. Okay, 10 isoprene units, so it's called Q10, right? And obviously you can buy Q10 in your you can go to Costco and you can buy Q10 and you can buy a big ass bottle of Q10 and it says good for your health. I mean, good for your heart. I think it's supposed to be good for your heart. Okay, cytochromes. Cytochromes, you have seen things that are related to cytochromes before. That is hemoglobin. Okay, so essentially what it is you have a heme molecule. This guy is a heme. Okay, it's a heme molecule. And this heme molecule will have an iron inside it. Okay, so just like hemoglobin. Okay, and essentially what you have is you have a protein that binds to the heme, okay, that covalently binds to the heme. Now this thing is a cytochrome. Okay, so the functional part of the cytochromes is essentially the heme and the iron. Okay, that's the part that is going to do a lot of function. Okay, and of course there are different cytochromes. For example, this is the general structure for cytochrome C. Okay, and you have a different structure for cytochrome A and A3. Okay, now cytochrome A and A3, instead of iron, it has a copper ion. Okay, but copper can also do oxidation and reduction. So iron, Fe, can go from +2 to +3. Copper can go from +1 to +2. Okay, but pretty much the same thing. Okay, for our purpose, they can take 1 electrons and donate 1 electrons. Okay, now what you should notice is that cytochromes, because it has heme, okay, because cytochrome has a heme group in it, it's going to absorb light. Okay, because hemes have a extended conjugation system inside it. Okay, so for example, your hemoglobin is red, right? Okay, so cytochromes a lot of times also have color. So you can essentially use a spectrophotometer to look at the absorbance of the cytochrome. Okay, and of course, if you look at the cytochrome, for example, here in this graph, these 3 graphs, you have cytochrome B, cytochrome A and A3, and cytochrome C. Okay, and obviously they have different peak of absorption. Okay, what you should notice is that the oxidized form and the reduced form, they have a different absorption pattern. Okay, this is obvious because whether or not you have 1 electron on the molecule, of course, cause the molecule to absorb at a different light wavelength. Okay, that makes sense. That makes sense. Okay. All right. So these proteins also differ slightly in their microenvironment to such that each heme will have a different property. Okay, and by property here, I usually mean that their reduction potential could be slightly different. Okay. All right. So if we take an even more closer look at the electron transport chain now, what you should notice is in complex 1, there are these different iron-sulfur clusters. Okay. In addition, before the iron-sulfur clusters, you have FMN. Okay, because you have to somehow change NADH from 2 electrons to FMN, FMM to this guy is 1 electrons. Okay. All right. And what you should also notice is that these enzymes are huge. All right. 1,000 kDalton. Yeah, that's pretty big. Right? That's pretty large. All right. Okay, so it's actually quite large. Okay, it's 1,000 kDaltons. It actually has 45 polypeptides inside. Okay, so it's a complex. And when we come back from the break, what you'll see is that it's this weird-ass looking thing. And then when things pass through it, it changes conformation. Okay, so complex 1 is pretty large. Complex 2 is slightly smaller. Okay, it's significantly smaller. Okay, and then complex 3, complex 4 is also smaller than complex 1. Okay, that is because it has less iron-sulfur clusters inside. Yeah, but complex 1 is actually quite large. Okay, but regardless, what I want you to focus on, okay, what you should focus on is the fact that you have different cytochromes and iron-sulfur clusters within the complex. Okay, for example, you should remember that complex 1 is FMN and then iron-sulfur clusters, okay, until it goes to Q. And then complex 3 is Q come in, and you have cytochrome B. Okay, so those are sort of functional proteins in complex 3. And then cytochrome B passes to iron, and then iron goes to cytochrome C. Okay, cytochrome C can then donate its electron to complex 4, which donate to a copper, and copper is probably typically going to be some kind of cytochrome. Okay, so cytochrome A, A3. Okay, so copper is in cytochrome A. Okay, so that you should know. All right, so cytochrome C has iron, but cytochrome A has copper. Okay, and in complex 4, it's going to pass the electrons around. Okay, eventually passing the electron onto O2, you generate water. In all of these processes, H plus, H plus, H plus. Make a note, complex 2 does not pump protons. Okay? All right? Complex 2 does not pump proton into the intermembrane space. Complex 1 does, complex 3 does, complex 4 does, complex 2 does not. Okay? This is a very important feature. It will allow us to discuss why FADH2 can generate less energy than NADH. Okay? So here we look at Q. Okay? This guy is complex 1. This guy is complex 2. Okay. Turns out, there are actually a bunch of other flavoproteins, okay, including when we talk about fatty acid degradation, we'll talk about this, okay. One of the enzymes in fatty acid degradation will put the electrons from fatty acid onto this reduced ETF. ETF, by the way, here, ETF, it stands for electron transferring flavoprotein. Okay, it's a long ass name, but what it is it's a protein that has FAD inside. Okay, FAD inside. And what that protein can do is it can directly associate to the membrane, okay, and then pass the electron from that protein to Q. Okay, so turns out there's a lot of different enzymes that can pass electron to quinone. Okay, and then once you get electron to quinone, then you're doing electron transport chain. 好吗? Okay. Complex 1. Okay. So what you should be wondering then is, let's say in eukaryotic cell, okay, in eukaryotic cell, glycolysis occurs in the cytoplasm, okay, cytoplasm, or cytosol, okay. You get glycolysis, and then you have NADH as a product, right? Glycolysis can give you how many NADH? Glycolysis can give you how many NADH? 2, okay? It can give you 2 NADH, right? So because glycolysis gives you 2 NADH, obviously you have to use that NADH. How do you use that NADH? Probably you're going to want to use the electron transport chain, okay? But, okay, here's the but, okay? Here's the but, not ass, okay, but, okay? What happens is that your electron transport chain is not in the cytosol. Alright? What do you do? Okay? Turns out you're going to use this Mallet shunt or Mallet shuttle. Okay? So what this guy is doing, okay, is essentially, you use a pyruvate plus NADH becomes Mallet. Right? Now Mallet has a transporter that can take you into the mitochondria, which it then split into pyruvate. Right? Why is it that NADH cannot just diffuse in and out of the membrane? Because it's too big. Okay? It's like huge. Molecular weight for NADH is like 800 grams per mole. Right? It's quite large. Mallet is like maybe 100 at most, like 100 something. Okay, 100 something. So the size of the molecule is different. So here you actually use a Mallet as a trans. It's like the truck driver, right? Or the truck, right? Okay, we're good. Now, and of course in the process, your complex 1 will spit out protons. Okay, now this is a confusing chart. Okay, so I'll just very briefly talk about it. It's not too important. Okay, but essentially is that what this chart is trying to show you is that when we do research, a lot of times for example, we know now the ETC looks like this, but how do we know it beforehand? Like how do people find out about this? So turns out we can use different inhibitors that will inhibit the ETC. For example, you can have inhibitors right here that can inhibit, for example, complex 1, inhibitor right here that inhibits complex 3, or inhibitor here that inhibits complex 4. Okay, so you can add inhibitors to the cell or to the mitochondria, and you look for the effect. For example, do you get an accumulation of a particular cytochrome? Okay, I'll talk about how we look at the accumulation of cytochrome in just a minute. Okay, but you can add inhibitor and you observe what happens. Okay, makes sense, right? It makes sense. It makes a lot of sense. Previously, we didn't have gene knockout methods, so doing knockout may not be easy, but nowadays you can just do knockout and you study. You study the effect. Okay, so additionally, what you also have is artificial electron acceptor. This is actually very useful for a lot of different purposes, not just to study roles of these electron transferring species, but sometimes also for engineering, for example. Okay, these things have a known standard reduction potential because they are chemicals. Okay, these are chemicals that we can use in the lab, and because they're chemicals that we can get, we know exactly what's their reduction potential. Okay, now sometimes, for example, if I were to study, let's say, what is a redox potential in complex 1, for example, okay, then what I can do is I can feed different artificial electron acceptor and see if they interact. Okay, because obviously, you know the value of the electron acceptor for this guy, okay, for this guy. Then when you feed it to the ETC, you can see whether or not this guy can be reduced. Okay, so that's how it works. All right, so you can use these different kinds of synthetic electron acceptors. All right, this other thing is how we study, is one of the methods of how we can study electron flow by interruptions. Okay, you can use this thing called the difference spectrum. Okay, and what this is that first you generate a spectrum by having a difference spectrum between a known reference measurement and a sample measurement. Okay, so in the example here, and looking at the black line, we have the black line. What it is it's a difference spectrum between a fully oxidized and a fully reduced ETC. Okay, so what does that mean? Okay, what does that mean? All right, difference spectrum, 你知道什么意思? Difference spectrum. So let's say I have one spectrum that looks like that. I have another spectrum that looks like, I don't know, looks like this. The resulting spectrum will look like that. Or, okay? So essentially, spectrum 就是 UV visible absorption 嘛 ，OK？ 那你 absorption 拿来，然后你两个 value 加在一起嘛，或是互减，应该说互减，减掉 ，OK？ 那你减掉的时候，你就会得到一个 difference spectrum。Make sense? Okay, so 就是他们的 difference 这样。Alright, so what you can do is you take a fully oxidized ETC. Okay, how can you get a fully oxidized ETC? 什么是 fully oxidized 的 ETC? Every component in the ETC is oxidized, right? How do you do that? Well, maybe you don't have NADH and you have a bunch of oxygen. Okay, so no NADH, so you have no source of electron, but you have oxygen, so oxygen will drain away all electron. 懂意思吗 ？OK， 懂意思哈？什么意思？什么意思？什么意思？好，来，ETC 嘛，你有 complex 1, 3, 4，然后最后到 O2， 对不对 ？OK， 好，来，你可以说，我不要给它 input NADH， 我不 input NADH， 但是我有 oxygen， 所以 oxygen 会把所有的 electron 都吸到 oxygen 身上，那你剩下的中间是不是全部都变成 oxidized？ 因为你没有 NADH 的 input。OK， 这是 fully oxidized 的 ETC。fully reduced 呢怎么弄？那你就有 NADH， 然后没有 oxygen 嘛，就好了，对不对？就是你没有一个 terminal electron acceptor， 但是你有一个 input of NADH， input of electron source， 那它就会变成全部 reduced。Make sense? Okay. Alright. So you take the difference spectrum of the fully oxidized and the fully reduced. Okay. You will get something that looks like that. Okay. And then what you can do is now you're studying, for example, what happens if you inhibit a particular complex, okay? In this case, you add this entamycin. Entamycin inhibits what? Entamycin inhibits complex 3, okay? So it inhibits it, all right? So you inhibit the transfer from cytochrome B to cytochrome C, okay? Therefore, you should observe an increase in what? In cytochrome B. 来咯 ，OK， 来咯，有没有？你这边有这个 chain 有没有？这是你的 ETC 嘛，哈，好来，我加了这个 entamycin， 所以就啪，我从这边切一半，那这是这个 cytochrome B 到 cytochrome C 这边断掉，所以这边都没了，那这边没了什么意思？那这边会变成 oxidize， 那但是这边呢会变成 reduced， 对不对？好来，所以你这时候呢，你去看它，然后你就会看到一个这个 cytochrome B 的 peak， 所以你就知道说，我会累积 cytochrome B。 Something like that, okay? Right? And you also disappearance of cytochrome C and cytochrome A. 为什么 cytochrome A 在这边 cytochrome C 在这边，这些就没了嘛，对，就是它不会被 reduce 了。Okay? Make sense? Right, so this is just a method to measure like where are you inhibiting. Okay, so that's just how it is. Okay, so this technique is useful for determining the effect of electron transport of a particular site on the ETC. Okay, now we're getting to the actual details of each complex. Okay, so we talked about the pathway level. Now we're going to talk about the protein level. Okay, so protein level, this guy is complex 1. Okay, all right. Now, it looks like an L-shaped thing, okay? It doesn't look exactly pretty, right? It's pretty ugly, but whatever. It's an L-shaped thing. Essentially, it has a leg that's sticking into the matrix of the mitochondria, okay? So why is that important? Because you need to have a soluble portion of the enzyme that can interact with NADH, okay? Remember, NADH is soluble, okay? NADH is not in the lipid phase. Okay? NADH is found in the aqueous phase. So you obviously need to have a portion right here that's going to interact with NADH. Okay? Now, once it interacts with, so this arm is what we call a hydrophilic arm. So hydrophilic arm. And then you also have a hydrophobic arm that's buried inside of the lipid. Okay? Now, the hydrophilic arm will interact with NADH, turn NADH into NAD, and then put the electrons onto FMN. Okay, now FMN. Okay, so let's look at this particular graph right here, the C graph. What happens is that, check this out. You have all these iron sulfur clusters. These are all iron sulfur clusters. Okay, you have a bunch of iron sulfur clusters. Okay, why do you need to do that? Like, why do you think you need so many iron sulfur cluster to do this thing? Why can't it just NADH come in, FMN, Q? Where is coenzyme Q? Coenzyme Q 在哪？在外面逛街，在家打电动。在 membrane 里面 okay, coenzyme Q 在 membrane 里面 so it's right here. Q is like right here, or Q is right here, okay? Q is in the membrane. So, if your FADH, your FMN is right here, how do you physically, okay, 我说 physically, how do you physically get electron from right here to right here? That's a long distance. Okay, long distance. Okay, so you cannot possibly, throw your electron across the membrane, ends up at Q. You can't do that. Okay, so what you have to do is you have to pile up a bunch of these iron sulfur cluster so that the distance between the two molecules is short enough that they can do a chemical reaction. Okay, so you have to be close enough to pass the electrons. 合理吧哈 ？Make sense. 你 physically 要够接近嘛，不然怎么弄，对不对 ？electron 不能飞出去。Alright, so if they're physically close to each other, then you can pass the electrons. Okay? Alright? Now, when you finally get the electrons onto Q, okay, so as the electron passes to Q, you have these enzymes called the NUOHK. You don't have to memorize that, okay? You just have to know that these are functional functional subunits of the complex 1. That is going to change conformation, okay? And you will cause an alpha helix to tilt the neighboring alpha helices and enable proton movement across the membrane. Okay, so all you have to know is that just remember when the electrons pass from your FMN to essentially Q, okay, Q is right here. Okay, Q is right there. When it passes to Q, you will cause a conformation change of complex 1. Okay, conformation change. Because the conformation is changed, you actually now have this channel. You have this channel right here. Oh, that's ugly. You have these channels right here that will essentially open, and when 2 protons passes through, okay, well protons passes through, then it will close again. Okay, so it's like a little gating process. Erase 掉手势. Okay? Alright? Make sense? So as the electrons pass from FMN to Q, you will cause a conformation change to the enzyme such that it now enables proton go from the matrix to the inner intermembrane space. Okay? And actually the proton has to pass through in order for the enzyme to pop back and then continue the next round of catalysis. Okay? Now, if we compare bovine, 你们知道 bovine 是什么 Yeah, okay, 反正 bovine 就牛啦 okay, bovine, cows, okay? So the bovine complex 1 looks like this. E. coli complex 1 looks simpler, but essentially the same shape. Okay, so this is just telling you that complex 1, even though between eukaryotes and prokaryotes, is quite different in their composition, but the shape is very similar. And the shape is important because you have to have a hydrophilic arm to grab NADH and a hydrophobic arm that grabs Q. Okay? Complex 2. Complex 2. What does complex 2 do? Complex 2 is SDH, succinate dehydrogenase. Okay? So in the matrix, you're going to do an oxidation of succinate to make fumarate, and you will pass the electrons onto FAD. Okay? And then FAD, which is, I can't even see it in this map, but essentially what FAD can then do is, this guy is FAD. FAD passes the electron to a iron sulfur cluster, which then passes to another iron sulfur cluster, which passes to another iron sulfur cluster. You don't have to memorize this, like is it 2 iron, 2 sulfur, or 4 iron, 4 sulfur. You don't have to memorize that, okay? Just know that you have different iron sulfur clusters. Each iron sulfur clusters cannot be too far apart, because if they're too far apart, you cannot do electron passing. So you look at the distance. They're about 10 angstroms, so about like 1 nanometer, close to 1 nanometer. So that's the distance which you can do chemical reaction. Right? And eventually, it also ends up in Q. Okay? So complex 2 also passes electron from FAD to Q. Notice, no protons are pumped. Thank you. Okay? All right, so Q is reduced to Q2 near the membrane matrix interface of complex 1 and 2. Well, what does that mean? Okay, what does that mean? Human animation. Okay, it's right here. Q is actually getting reduced between matrix and the intermembrane space right here. And then once it's reduced, it flies into the membrane. Okay, why is that important? Okay, because Q becomes what? Q becomes QH2. Okay, quinone become quinol, right? And once you add quinol, it's QH2. You have 2 protons, right? Where do the protons come from? You have no protons in the membranes. Okay? 你在 membrane 里面你不可能有什么 proton 在那边飘嘛，好不好？可以吧？应该可以理解吧？在 membrane 里面你不会有 H+ 在那边飘，因为它是 hydrophobic. Alright, so Q is actually getting reduced around the membrane matrix interface so that it can grab 2 protons from the matrix to become QH2. Okay? Alright. And then QH2 is going to like move around and then until it hits complex 3. Okay? And complex 3 looks like this. Right? So yeah, obviously here I give you 2 figures. One is the ribbon figure. The other one is more like a cartoon drawing. Okay? So essentially, Q will come in, right? And essentially what you'll do is you'll do this process called the Q cycling. Okay, I'll show you what Q cycling in just a minute. But the net reaction is that you will release 4 protons to the intermembrane space from QH2. Okay? So you actually do 2 QH2 for every 2 electrons transferred to cytochrome C. Alright, so like this. Alright, so it's like this. Oh, this is so complicated. Alright, it's actually a lot easier than you expect, okay? But let's not look at the bottom. Let's just look at what happens first, okay? The Q cycle essentially has 2 stages. Right? 有2个 stage. The first stage, 或2步啦，应该这样讲. First stage, you have a QH2, right? 所以这是complex 1, 然后会吐出一个 QH2, okay? And then QH2 will then be split into homolytic cleavage. Okay? So 1 electron goes up, 1 electron goes down. Okay, so homolytic cleavage. Okay, because QH2 has 2 electrons. 可以哈? 2 electrons. And then one of that electron, okay, passes to cytochrome C. Okay, cytochrome C is the end product of complex 3. Okay? So one of them ends up at cytochrome C, which is fine. You're happy. Okay? The other electron, okay, will actually go and interact with a Q, another Q, not QH2, 一个 oxidized Q. And then you make QH radical. 可以吗? Alright. Stage 2, you have another QH2 that comes in and it becomes Q. Another homolytic cleavage. Okay, you cut it out. Okay, and again, 1 electron goes to cytochrome C. Another electron goes up. Now, however, this time the Q radical can come in, react with this electron, and you form a QH2. And then QH2 can do this. Okay, so this is why it's called Q cycle. So this is how Q is able to pass 1 electron at a time to cytochrome C, because Q itself has 2 electrons. So then you want to pass, every time you pass electron from QH2 to cytochrome C, you can only pass 1 electron. So that's going down. Okay, but as a result, you will always kick out 1 other electron, and that other electron has to go somewhere. You can either use Q to take that electron to make Q radical, or you use Q radical to take that electron and become QH2. Okay, makes sense? But regardless, the net reaction per QH2 is that 1 QH2 give you 2 cytochrome C reduced and 4 protons into the intermembrane space. And how does the proton go into the intermembrane space? Because QH2. Okay, this is why 我上一张 slide 说那个 Q 被 reduce 的时候，在那个 membrane 跟 matrix 的边界，这很重要，因为那个 proton 啊，它变成 QH2 的 proton 是 matrix 的，但是吐出去的时候是吐在 intermembrane space， 所以 Q 本身就是带一个 proton 的卡车啦。OK， make sense？ 懂我意思吗？有没有听不太懂？可以哈，可以啦，可以。 All right, complex 4 is somewhat easier. Complex 4 is essentially you have cytochrome C comes in and interact with this guy's complex 4, and essentially what will happen, this guy is cytochrome C, right? Cytochrome C comes in, pass the electrons, okay, to copper, to another copper, to another copper, and then essentially you will interact with O2, come in, and okay, you have a substrate proton on the matrix side, okay? 在 matrix 那一面，你拿4个 proton 去跟 O2 结合，然后你形成 water. In the process, you also then essentially pumped 4 proton over to the intermembrane space. Okay? Alright, so the net is 2 protons are translocated for every 2 electrons transfer. So 4 protons for 4 electrons. Okay? Alright, so turns out this is the model proposed, the supercomplex of complex 1, 3, and 4. Turns out 1, 3, and 4, they're really close to each other, so that Q doesn't have to travel for a very long time. And then cytochrome C also don't have to travel for a very long time. Okay, so that complex 1 immediately after Q becomes QH2, immediately goes into complex 3. Complex 3 passes the electron onto C, into cytochrome C. Cytochrome C moves slightly, okay, and then it goes to complex 4, and then it will then pass the electrons up to oxygen, generate water, okay, and then you regenerate the cytochrome C, oxidized form, and then continue the entire process. Okay, so the mitochondrial electron transport chain is composed of these enzymes, right? And their prosthetic groups are listed here. Okay. Cytochrome C is not an enzyme, it's just a protein that carries the electron. Okay? Alright. Now, proton gradient. Okay, so now you created this thing called proton gradient, separation of protons. Okay, you have a lot of protons in the intermembrane space, not so much proton in the matrix. Now you have a chemical potential. Okay? And so this summarizes complex 1, pump 4, complex 3, pump 4, complex 4, pump 2, per 2 electrons. Okay, this is per 2 electrons. Okay? Right, so you get a total of 10 protons for using complex 1. You get a total of 6 protons per using complex 2. Now, to make ATP using the ATP synthase right here, every time 4 protons, actually the number is not exactly 4, but we usually just say 4 because 4 is a good number. It's a round and even number. So for every 4 protons that go from the intermembrane space, come out, okay, back to the matrix, you will generate 1 ATP. Okay, so therefore, we can define this called PO ratio. What it is essentially you take how many protons that you can generate, okay, per using essentially 1 molecular oxygen, so 1 ATP. Okay, so to generate 1 ATP, you need to have 4 protons. Okay, 4 protons. But for NADH, you can generate 10 protons. Okay, so if you divide that, you get 2.5 ATP from NADH, 1.5 ATP from FADH2. This number you have to memorize. Okay. So now we finished talking about ETC. Any questions on ETC? Okay, so the remaining 10 minutes, I'm going to talk about ATP synthase. So because you have this separation of protons, we can call this thing the proton motive force. Note, it's not a force. It's a potential energy. Okay, so but of course back in the days, what do these people know? Okay, so they call it a proton motive force. Right? It's a potential energy. So this is the potential energy across the membrane. Okay, now because the proton pumping from complex 1, 3, and 4 a proton gradient or electrochemical gradient is formed. Okay? This separation of charge or difference in pH provides the driving force necessary for ATP synthase. Okay? And typically the pH difference between matrix and intermembrane space is about like 0.75. It's not a lot, but if you think in terms of pH is a log scale, is quite large. It's close to 10 times the difference. Okay? So this guy somewhere, he then did a calculation where he can show that for every proton transfer back into the matrix, the free energy of this process is about minus 21 kilojoules per mole. Okay? Therefore, about 210 kilojoule per mole of energy is stored in this electrochemical gradient, as 10 H plus are translocated in the process of NADH oxidation to O2. Okay? Remember how much energy is released for NADH oxidation to O2? 210. Right? So it's about the same. Right? So the energy from NADH oxidation to water, okay, so NADH passing electron to O2 making water, you will store about 210 kilojoule of energy. Okay? Now, only 2.5 ATP is formed, so where's the rest of the energy, right, to provide driving force? Okay? This is ATP synthase. Okay, so ATP synthase is a doorknob. Okay, it's literally a doorknob. 你知道 doorknob 是什么？门把嘛？那个圆圆的那种门把，那个叫什么？喇叭锁那种形状，就是它在那个 membrane 这边呢，这一块啊不太会动，上面会动，上面会 rotate. Okay, so essentially ATP synthase is composed of two complex. Okay, two complex. We call it F0, F0 and F1. Okay, top is the F1, bottom is the F0. Okay. Essentially, what you have is F1 knob. Okay, doorknob, F1 knob that can rotate, and that's inside of the solution. So this guy is in the matrix, and it's connected to a central stalk to the F0 base, which is this part doesn't move. Okay, and then this thing moves, but this thing doesn't move. And what happens is your proton movement is going to go from that direction. Okay, now you don't necessarily have to remember the details of ATP synthase. However, I do want you to remember that you have F1 knob contains 3 AB dimer. This is a very important key. Okay, it has 3 parts. Okay, so the doorknob is split into 3 parts. Okay, and basically here it just says that the base, okay, the stalk here, the F0 base, is composed of a bunch of C units, okay, and a peripheral stalk that's composed of B, D, F6, and blah, blah, whatever. Okay, doesn't really matter. Okay, now, the central stalk in the C ring is the rotor of the ATP synthase. So actually, this thing, C1, C10, this thing actually rotates. So this part. This part will rotate. Okay? This part essentially will rotate. Alright? Now, essentially what happens is this. Okay? Your proton is going to go from the inner membrane space, entering this shaft, and proton is going to move around. And remember, here's a electrochemical potential. Here's lots of low proton solution. So the proton naturally want to move in this direction. And as the proton move, this whole thing, this thing, is going to rotate. Okay? Does it rotate? Actually, this one doesn't rotate. This one, as it moves, it's going to rotate the top thing. Okay? Essentially this. Okay, what I want to show you is this. This is why you have to know the alpha-beta dimer. Okay, so on the doorknob, okay, on the alpha-beta dimer, okay, you have 3 sites. Okay? And these 3 sites, we usually call them loose, tight, and open. Okay, so what happens is this. Okay, what happens is this. Okay, it was observed in the tight position when ADP plus PI to ATP has a delta G of 0. Okay, so that's weird, right? Because typically, if you want to make that amount of energy, that should be plus 30. Okay, but in this case, it's 0. And this happens because ATP is actually bound on the enzyme because the enzyme has a strong affinity towards ATP. That's higher than ADP. So if the enzyme has a very high affinity for ATP, ATP is stabilized, okay, so that ADP become ATP is okay. Yeah, it's actually easy. Okay? Now, so turns out, okay, as the hydrogen, the H plus, the proton, rotates the knob, okay, the enzyme will change conformation from tight to open, okay, then the binding affinity for ATP will decrease, and then ATP can then release. Okay, so it works like this. Okay, so the gamma position, this thing is going to rotate. Okay, this thing rotate together with the thing in the bottom, and as it rotates, the alpha beta dimer changes conformation. Okay, it changes conformation. All right, now in the tight position, so this guy is the tight position, ATP is stable. So at this particular stage, ATP can become, ADP become ATP. Okay, so it's at this stage that ATP is formed. Okay, because the energy is close to 0. Right, and then when it rotates again, then ATP binding affinity drops, so ATP flies out. Okay, so ATP flies out. Okay, so if we only look at this guy. Okay, what happens next? When you rotate again, it's going to become the this particular one doesn't change because you're changing the conformation of this. Okay, and here what happens is that your ADP and PI goes into the open state. Okay, so here is a rotation, here is just like that, and here is another rotation. Okay, rotate. Now ADP and PI is now in the loose state. Okay, so it captures the ADP and PI. Right, and then when you rotate again, it becomes the tight conformation. Okay? Does that make sense? So you keep on doing open, loose, tight. Okay? In the open scenario, ATP come out, ADP come in. In the loose scenario, okay, ADP and PI is there. When in the tight, okay, ADP become ATP, and then when it pops open, ATP comes out. Okay, so the reason why the conformation can change is because that the proton is moving. Okay, proton is traveling through this whole thing. So this thing rotates. Okay? It physically rotates, and because it rotates, it causes the gamma subunit to rotate. And when that rotates, it changes the conformation of your alpha beta dimer. Okay, and alpha beta dimer is changing open, loose, tight, and then that will promote ADP binding, change conformation, ADP binding become weak, ATP binding increase, so you actually favors the formation of ATP. It becomes open, ATP comes out, okay, and then you can generate ATP that way. Does that make sense? This is ATP synthase. So it's literally like a little doorknob. All right, so if you do an in vitro study on it, what people have done is that you actually show the rotation of the gamma subunit. So you can essentially purify the alpha beta dimer, okay, and have them attached to a nickel NTA bead. So this is using his tag, okay, and what you can do is you can actually feed ATP, so the reverse reaction, okay, the reverse reaction of the natural process. You feed ATP, and ATP hydrolysis will cause it to rotate. Okay, you can actually rotate this thing. You fuse a actin filament on it, and you label the actin filament. You put ATP in it, and you can show that ATP synthase will actually rotate in the other direction. Okay, so it's shown right here. Okay, does that make sense? Right. So on my slide, you have some ATP synthase animation. You can go check it out. Right. So when we come back on Tuesday, I will sort of finish up ATP synthase, and then we're going to be talking about some minor stuff that happens in mitochondria, okay, including how you combat oxidative stress. Okay, and then that's it.